Hi guys, welcome back. Today we're going to be doing Tools of the Biologist for your class notes. So you're going to refer to pages 2, 3, 4, and 5 in your class notebook. So ideally you're going to the OneNote, you're going into your class notebook and you're going to type the notes right in there. Although if you were in class with me, you received your note packet. And if you would like, you could write it in the note packet first and then transfer it over to the class notebook. But please get into the habit of using the class notebook. If you are confused with how to use it, you can watch the YouTube video that was posted on. It's like a 6-minute video that was made by one of my colleagues, Mr. Greb, who's a science teacher in Ross, and he made a video on how to use the OneNote. So today we're going to go through the notes, again, pages 2, 3, 4, 5 in your class notebook. Okay, so let's begin with Tools of the Biologist. So this is broken up into categories. The first section here is the measuring tools. So there were 4 types of measuring tools that you're going to put into your notes. So you can feel free to pause the video if I'm going too quickly, but I'm going to go through each slide. I only have 15 minutes to record my videos because then I get cut off. So I'm going to be kind of going a little quickly, but of course you can pause it at any moment. Okay, so the ruler, of course, it's used to measure the length of an object. You'll be familiar with most of these tools, so I think this will go kind of pretty quickly. The next tool is a balance. There's 2 types of balances, an electronic or a triple beam balance, but either way, it's used to measure the weight of something. So it accurately measures the weight of a specimen. The next tool is a graduated cylinder. It's used to transfer and measure liquids. It's more accurate than a flask, also known as an Erlenmeyer flask, but it's still used to transfer. The reason it's more accurate is it has a lot more numbers here and lines, so you could be a little bit more precise with your measurements. And the last part in this section is a thermometer, which measures the temperature or the amount of heat in something. Okay, that's the first 4 tools. Now we're going on to the next category, which is safety equipment. Again, you might know most of these already, so we're just going to focus on the ones you don't know later on. Okay, so safety goggles, of course, is used to protect your eyes. Anytime we use chemicals, we would need to wear safety goggles. Another piece of equipment for safety is an apron, and this is basically just used to cover your body and protect your clothing. The next piece of equipment that's important is gloves, which is used to cover and protect your hands. This is what I'll be using in the classroom anytime I hand out any dittos, which will not be very often, which is why we're using the digital notebook. And the last bit of safety equipment is called a hot hand. These are kind of like rubber little pockets that you slip your hands into like mittens, and it's used to carry a hot beaker. Okay, so moving on to page 3 in the class notebook, or if you have the hard copy and you're looking at the hard copy, it's page 3 here. We're going to get into the containers. Some of the containers that we're going to use in the lab are a beaker, which is used to hold fluids. So if you look at this picture to the left, all you're writing in your notes is it's used to hold fluids. But if you look at the picture, we want to know how much fluid is in this beaker. You can read the number here. There's 200 mLs of fluid there. So it's just kind of showing you how to read it. The next tool is a flask. Again, it's used to hold and transfer fluids. That's all you read in the notes. In this one, how much fluid is in here? Again, you read here. It's at the 125 mil mark. How much is in here? More than that, because this is where it was 125. So it's more than that. Not as accurate because I don't know what number that is exactly. That's why the graduated cylinder is more accurate. The next piece of equipment is a funnel. That is not in your notes. It's just something I'm explaining to you, but you probably use this in your home. I use it all the time when I'm filling my salt shaker. It's just used to assist in transferring liquids or powders or salt, things like that. A petri dish is the next one in your notes, and this is used to grow bacteria. So why would anybody want to grow bacteria? When you go to the doctor and they take a swab and they put it in the back of your throat and they put it on a petri dish, which contains something called agar, which is a nutrient. If bacteria grows, then it means you have a bacterial infection. So it's used to check if somebody has an infection or if there's bacteria on a surface. The next piece of equipment is a test tube. A test tube is used to hold liquids. The next and last in this category is a test tube rack, which is just used to hold the test tubes. So again, pretty easy. You know most of this equipment already, I'm sure. Okay, moving on to the next category, which is the holders. This is known as a test tube holder, which is used to transfer a hot liquid that's in a test tube. So normally you could just carry the test tube, but if it's hot, you don't want to burn your fingers, so you need a test tube holder. If you want to carry a beaker, the test tube holder is too small for that, so you need these, which are called tongs, or you could use the hot hands from the previous slides. Again, it's used to transfer hot objects, mainly a beaker or a flask. The next holder would be a forceps. Back in the day, it was like not scientific enough to call it a tweezer, but now it's acceptable to call it tweezers or forceps. But basically, they're used to pick up small objects, to move small objects, or to pick them up. And the last thing in this category is a pipette, which is used to transfer fluids, mainly small amounts of fluids, on like 1 drop at a time. Okay, the next category is your dissecting equipment. So this is the dissecting tray, which is where you'd place the specimen during your dissection. These are pins, dissecting pins, which is used to hold the object in place, so when you cut it, it doesn't slip and cut you. This is also called mounting. So when you're mounting an object in place, it's just meant to keep it safe so it doesn't move around. The next thing is called a probe, which is just used to assist during dissections. You might want to be showing a student something and point to it or move something around. They can be perfectly straight or straight, like with a little curve at the tip here, like this one. And the last thing is the scalpel, which of course is very, very sharp, used to cut your specimen. Whenever you're doing a dissection and you're cutting, you always cut away from you or anyone else in your lab group. Okay, so we're turning on now to page 5, which is your lab techniques. This is the most important section of these notes. So this is where I would say, if you're in your OneNote, maybe change the color of your font when you're doing these notes, or put a little symbol next to it, just so you know this is the stuff that Mrs. de Blasi says is most important for today's lesson. And therefore, it's something I really have to focus on because it's something you never heard of and you don't know much about. So these 3 pictures show different lab techniques. Each of these techniques is used to separate something, depending on which tool you're using. The one at the bottom here, which is called gel electrophoresis, it's used for DNA. The one on the side here, it's used to separate colors. And the one at the top here is used to separate by density or weight. So let's start with the electrophoresis. It's used to separate chemical mixtures, specifically DNA, and it uses an electrical charge. That's why it's called electrophoresis. The smaller molecules will move faster. So this is called a DNA electrophoresis gel. So this is 5 individuals' DNA that was put into this gel, and they move up. If they're small, they'll move faster. So they'll move all the way up the gel. And if I was asking you which 2 individuals here are identical twins, you'd compare all the bands of these 5 individuals. This is individual 1, 2, 3, 4, 5. And you can clearly see that individual 2 and 3 are identical. So this is used to compare and separate DNA. The next technique is called chromatography, and it separates mixtures by size through the solvent, but mainly it's the color that it's separating. So if I had a droplet of, say, the color purple, and I put it into a piece of chromatography paper, it would start to separate out. If it's purple, it would be blue and red, because that's what makes purple. If it's another color, it would separate into different things. So it makes like a pretty little rainbow. It's just separating colors in that particular pigment. And the last technique is the centrifuge, which separates mixtures of solids and liquids by spinning it, and it separates by density. So here's the machine. It puts the test tubes in, and it turns really, 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 really fast. It spins super fast. And at the end of the spinning, it will separate. So in this case, the heavier object is the white part, and the lighter part is the yellow, and it separates by density. Okay, the next section is the microscope section of the notes. These are different types of microscopes. So we're focusing on the compound microscope. That's the only one that's going to be in your note packet. The compound microscope is used to magnify an object by using a lens or a combination of lenses and light. So here's the microscope. It has a mirror down here to reflect the light. And in this picture, we're looking at cheek cells. So if you extract cells from inside your cheek with a toothpick and put it on a slide, you would see this. The little dense part in the center of each cell is the nucleus. These are some other types of microscopes, but they're not in your notes. But this is a dissecting microscope. It's used for larger objects. You don't have to take notes on this one. And this is an electron microscope, which is used at NASA or universities to really magnify like 200,000 times. Again, that's not something we're going to use, so no need to take notes on that. What you do need to take notes on is the next part, which is a cover slip. So assuming this is your saliva, you would need a cover slip to place over the specimen so you could use it with your microscope. This sheet that's underneath it, this piece of glass under it, that's called the slide. And that's used to place the specimen on, and of course use again with the microscope. This is actually not in your notes also. It's just a magnifying glass, which is basically a simple microscope. It only has one lens, but it's used to observe something that's small and make it slightly larger. That's what I use my glasses for. My glasses are basically a magnifying glass. Helps me see something, since now I need glasses when I see something, when I'm trying to read something up close. The last thing in the notes is called resolution, and it's used to make an object clearer. You could see in this picture that the picture on the left clearly has better resolution than the picture on the right. It's called resolving power or resolution. Okay, guys, that's the end of the notes for today. Again, hopefully you're taking them in your in your class notebook in OneNote. And again, if you don't know how to do it, please refer to the YouTube video that I posted to watch that 6-minute video to show you how to do that. All right, guys, that's it for today. Have a great day, and I'll see you all soon.